Welcome to our discussion of the recent discovery of the COVID vaccine for the COVID-19, discovered in great part due to the amazing research of the last 20 years by Dr. Drew Weissman, who's married to Mary Ellen Weissman, who's the daughter of Morris and Ann Weiss, members of our congregation, our community. We're very honored to have Dr. Weissman and his wife here with us today. Dr. Weissman is the professor of medicine at the University of Pennsylvania in infectious disease, professor of infectious disease, and he's at the Perelman School of Medicine there at University of Pennsylvania. It was his papers that were able to get going with this mRNA type of vaccine. So we thank him for joining us today. Appreciate his taking this valuable time to speak with us. Thank everyone for joining us, friends, cousins, scientists joining us today. Great to have everyone, and thank our partners for today, Baron Hirsch Congregation, Rabbi Lehrfield, the Beth Shalom Congregation, that's Rabbi Sarit Horowitz, and also her husband, Avi. Welcome him as well. And also Kentor Julian, Rabbi Kentor Julian of the Dor Chadash and Senior Lunch Bunch from the Jewish Family Service. We welcome one and all to this great occasion. We'll get right to our questions. Our first question is, some people have all kinds of different autoimmune diseases. The nephrotic syndrome, the diabetics, thyroiditis. Some people have all these things together. They have no thyroid gland. So number one, is it safe? Number two, will it work? And people are worried about, what if they had lupus or other things, multiple sclerosis, all these different people. What's going to happen with them as far as this vaccine goes? Will it work? Is it dangerous for them? Dr. Weissman, please illuminate us on this question. Thank you.
We take vaccines, we take tetanus, we take flu, we take MMR, we take lots of different vaccines. All of them stimulate the immune system. We haven't seen lupus, MS, other outbreaks with other vaccines. There's no reason to think that this vaccine is going to be any different. We just don't know. We haven't studied it, so we can't say for sure.
One of the big concerns is to try to explain what happens afterwards. So you take this second dose, you have the booster, and then you have a 95% chance of not getting it, but you're still in danger of carrying it in your nose. Is that because you might have 5% possibility that you did get it and it's a mild case? Could you clarify what's the problem after you get the second dose, and what does it mean as far as mask wearing, going out, and normalizing life?
Sure. So we've got more data on this now. What we know is that after the first vaccine, you have between 50 and 80% protection from getting symptomatic infection. So that's pretty good. After the second vaccine, you have 95% protection against getting symptomatic disease, getting a sore throat, etc. You have 100% protection against getting severe disease, ending up in the ICU on high-dose oxygen. The Moderna study looked, and what they found is that they reduced asymptomatic infection. And asymptomatic infection is the big problem, because when somebody doesn't know they're infected, they can transmit, and people don't know, oh, that person has COVID, we need to stay away from them. Many people don't wear masks. If they have asymptomatic infection and they're not wearing a mask, they can transmit. What they found in the Moderna trial was that asymptomatic infection was reduced about 2/3. So that's good. It isn't perfect. We would have liked it to be 100% gone, but that's rare for a respiratory virus. What we still don't know is that 1/3 of people who had measurable virus in their nose, whether they're able to transmit. In the animal studies that were done before this vaccine, the amount of virus in the nose of monkeys and mice was about 100-fold lower than in an infected person. What we still don't know is that enough virus to transmit.
So when 70% of the people have it, will all those concerns go away?
So
Have the vaccine?
yeah, I mean, the ultimate goal is to reach a level of herd immunity. We don't know what that number is. It could be 70, it could be 80, it's something around there. The idea is that if we get that percent of the population, then even if somebody transmits, there'll be nobody for them to transmit to. It'll be 1 out of 5 people are unprotected. But if the virus, if each person only infects 3 to 4 people, and 1 out of 5 is capable of being infected, then the virus will die out.
I'll ask 2 questions. Number 1, any further guesses on how long this vaccine will last when we'll need another booster? And then another question, it seems that you could use this same type of thing, as you said, on other diseases. How far are we from using this type of vaccine for, I don't know, cancer or some other horrible kind of a disease?
Yeah, so we don't know about durability yet. We know about durability in mice and monkeys and chickens and rabbits and guinea pigs and lots of other animal models, and it's very good. But humans are a different thing. So until we have the data, we can't say how long it's going to last. Preliminary data says that it's fairly durable, so it will probably last years, but that's a guess. We just don't know yet. My lab has been working on this vaccine platform for 7 or 8 years. We actually have 5 phase 1 clinical trials that were planned before COVID hit. 1 was for genital herpes, 2 were for influenza. 1 of those was a universal influenza. 2 were for HIV. So we've been making this vaccine for years. We were ready to go into people. My guess is that in the coming years, the vaccine will be used for a lot of different diseases and a lot of other therapeutics.
Can cancer be affected with this as well, or it has to be a virus?
So we are doing studies on cancer as well. There are vaccines that are currently in use for cancer. These are often known as personalized vaccines. So what that means is you take a person's tumor, you sequence it, you measure all of the proteins looking for mutations, and then you make a vaccine based on each patient's cancer. And those vaccines are in clinical trials right now and have shown some success. So there is a future for RNA vaccines in cancer.
Were there a lot of naysayers to RNA vaccines? People who said, "What are you working on RNA vaccines? What is this? It's nothing. It's let's focus on other things." And were there people who were sort of saying, "We're not going to give you money for that. We're not going to give you grants for that"? Were the people skeptical about that?
So Katie Carrico and I started working on RNA about 22 years ago, and up until the past couple of years, we would hear that all the time. People didn't want to give us money. Biotech companies and pharmaceutical companies didn't really care what we were doing. They just had no interest in it. And then as the data kept getting better and as the results kept getting better, people became interested.
So what is the relationship between you as a professor at U of P and the pharmaceuticals? I mean, you just handed over all your research and then it went over. What's the relationship?
Yeah, so the pharmaceutical and biotech companies license the technology from Penn. So they have freedom to use it however they want.
Interesting. Now, one of the big questions people had was about the second dose. Here we're having very strange rollout. It's supposedly for 1A1 workers and then some other workers, but then again, sometimes they have some extra, so they put out a note that it's for older people can get it. So you get your first dose, you don't know if you can get your second dose. So what's your whole feeling about this relationship of first dose, second dose? I know in England, they're thinking of just using one dose and get everybody one dose. And then what if you end up getting your first dose, but you can't get your second dose on the official day when you're supposed to, and you get it much later? Does that mean you have to start all over again, or is it not a big deal? What do you say?
So the problem is we don't know. The clinical trials set up to give one dose followed by the second dose 3 or 4 weeks later. That's the only thing they studied. They didn't study what if you wait 6 months before the second dose. They didn't study what if you only give a single dose. So we just don't know what the answer is. What the Biden administration is saying is that we think we'll have enough new product coming in that we can give out everything we've got and wait for the new product to use as a second dose. They have a much better view and understanding of this than I do. I'm always nervous that I don't trust pharmaceutical companies to do what they say they're going to do.
We have just had a question. If people have COVID, how long should they wait before getting the vaccine?
We don't know that either. So we know that people with COVID can get reinfected. The documented number is low. There's been, 22 million people infected with COVID, and maybe 20 people have been reinfected. So that's incredibly low. The issue is that some people have found that the antibody response to COVID infection isn't durable, and it may wane over time. So our recommendation is that people that have had COVID should get vaccinated to boost their antibody response and give them better protection. How long to wait after COVID, we don't know.
There's some questions about shingles. If someone's getting a vaccine for shingles, should they get that before or after this? There's a question about kids. If kids are not going to get the vaccine, how's it going to affect the whole rollout? And what do you have to say about these allergic reactions that are happening in a few people during this time?
Yeah, so I apologize, but the answer to most questions is we just don't know, because it's a brand new vaccine. It's just been given out. We don't have studies that tell us what to do. As far as the shingles vaccine, I'd probably tell people to wait a week in between the vaccines, because both the shingles and the COVID vaccines give you arm pain and arm soreness that will last a day or two. And just to give you a break, not having a sore arm for a long time.
If someone's near surgery, can they take it a few days before surgery? Any comment on these allergic reactions that have been taking place?
So the only risk on surgery is that some people, not a lot, but some, will have fevers for a day after the vaccine. And if you walked into your surgery with a fever, they wouldn't take you. If you walked into the surgery with flu-like symptoms or any symptoms, they wouldn't take you for surgery. So if you do it 5 or 6 days, then you're safe. I wouldn't do it a day or two before surgery, because you just don't know if you would have a bad reaction. As far as the allergic reactions, what those are, they're called anaphylactoid. So anaphylactoid is different from anaphylaxis. Anaphylaxis is an antibody against an allergen, like peanuts or pollen or other things. Anaphylactoid means that something stimulated your body to make those mediators that give you those symptoms. They haven't found an allergic reaction to the vaccine that's causing these reactions. They're happening, the rates are around 1 in 100,000 people. So we consider that a really rare side effect.
I guess overall it's going fantastically well.
Yeah.
Yes. I have one simple question, then some more technical and geeky kind of questions. A simple one is right here from Ellen and Stan. Yeah, any word about the spoilage of these vaccines when they get out of the freezer? And if you don't have a sore arm, does that mean the vaccine didn't work on you, that your body didn't react properly?
No. So only 80% of people had a sore arm. What's interesting in looking at the pictures, what's hopeful is that older people have fewer adverse events. So they have less arm pain, they have less flu-like syndromes. We don't know why, that's just how it is. So not having arm pain isn't a bad thing. As far as spoilage, right now the vaccine is only being given out by health centers that have adequate faculty, staff, and refrigeration. So they're not going to let that happen. The issue is what happens when you start shipping this vaccine out to CVS or Walgreens or whoever your local pharmacy, and will they follow the same recommendations or requirements for giving out the vaccine? I have nothing to do with that, so I don't have an answer on how that's going to be policed.
And why does the Pfizer need to be stored at such extreme cold conditions?
Yeah, so the issue is that what the vaccine is, it's mRNA that's put into a fat droplet. That fat droplet has a specific structure. To maintain that structure, you have to freeze the fat below a certain temperature. And that temperature, it varies, but it's around -40 centigrade. And they rushed to make this vaccine, so they kept that. They did work to keep stability at 4 degrees in a refrigerator for up to 7 days to make it easier to give the vaccine out. Moderna spent more time on this, and they figured out how to stabilize the lipid nanoparticle, the fat droplet, so it would be stable for months at -20, which is standard freezer temperatures, and I think up to a month at 4 degrees. Pfizer is now going back and redoing those studies and that development so that their vaccine will hopefully someday be stable in a regular freezer and stable in a fridge for weeks to a month.
Gotcha. Now some questions that seem to come from some scientists. Maybe you know them, Paula Miller and Ehud Kamin. One is from Paula. Is there a specific lipid nanoparticle that is used as a delivery system, way above most of our heads? Number two, which cells does the mRNA go into to produce the protein? Are those cells attacked by the immune system? If so, why do we not see necrosis at the injection site where there's a higher concentration of mRNA into the cells?
Yeah, so the lipid nanoparticles that are being used are the same construction, the same type of particle that's part of an FDA-approved drug for a genetic amyloidosis. The lipids are a little bit different. They've been optimized for mRNA, but that's the general construction. It's 4 lipids are mixed with RNA, and they form a particle. The cells that take up the mRNA are varied. And what actually happens, so if you inject them intravenously, they go to the liver and they're taken up by hepatocytes. If you inject them into the muscle in your arm, they're taken up by all of the local cells. The cells that take them up the most and make the most protein are the dendritic cells. What the dendritic cells do, they migrate into the muscle after the injection because of the adjuvant activity of the lipid nanoparticle. They take up the particles, travel to a lymph node where they stimulate the immune response and they make the antibody and T cell responses. And they keep doing that for about 2 weeks. We don't see any immune attack at the site of injection. That's probably because the amount of protein made by muscle cells is very low and there's nothing to attack.
Okay, there'll be an exam on that for those who are participating afterwards. But I'm sure many of the scientists and doctors on here really appreciated that answer. So thank you so much for that. Firstly, I just kind of want to give you a tremendous bracha for everything you've done. I mean, how amazing you're saving. Who knows how many millions of lives, and it's just amazing. And we're so honored that you joined us today. That to even know you and have an association with you through the whole Weiss Katz family here in Memphis and Elster and everybody else. Let's see, a couple of last minute questions. Yes, you mentioned 95% after the second dose, but there is a couple week lag there to get to 95%. Is that right?
So it's 10 days after the second dose that you see the full 95% efficacy. After the first dose, up to 80% of people can be protected. So you're going from 80 to 95, not from 0 to 95.
Someone wanted to know, someone named Lab, wanted to know if after your vaccination, are there tests to tell if you are immune?
Sort of. There are antibody tests that are used to measure, did you have infection? One of those tests, or one set of those tests, measure spike antibodies. They're not quantitative. So what they tell you is, yes, you have spike antibodies, or no, you do not. They don't tell you, do you have enough to protect you? So they're really not that useful.
Okay. Cousin Jennifer wanted to know, Jennifer Roberts wanted to know if, just lost her comment. She wanted to know if you could get the second dose a little bit early. Like maybe there's a surgery they had coming up later. They want to get done with the side effects. So they're gonna, instead of waiting 2.5, 3 weeks, they're gonna go early for the second one. Does that make any sense?
Yeah, so I can't answer that. She has to ask her physician about that.
Gotcha. All right. Any other questions? Please put them up there. What is the maximum time after the first dose the second dose can be administered and still achieve maximum benefit?
Yeah, so what was tested in the phase 3 clinical trials for the Pfizer, I think it was 24 days. For the Moderna, it was 32 or 33. That doesn't mean that it won't work if it's given a month later. We just don't know.
Okay, and I hear that your vaccine works for, these vaccines work for the other COVID permutations and mutations, right?
Well, we think so. People have to realize that this virus came over from bats in November or December of the year before last. That was the first time that we know of that it ever went in humans, which means that the virus has to figure out how to better infect humans, because it had infected bats for probably thousands of years. So that means the virus is going to mutate, and it's going to figure out how to infect humans better. So there have been a lot of mutations that have appeared over time. And so far, all that we've looked at are still susceptible to the vaccine. We haven't tested the current UK and South Africa strains that are circulating throughout the world, but we believe that they're also going to be sensitive to the vaccine.
Great, fantastic. All right, let's see. What else? see. Um, second dose measured. Yes. Any other oral questions? I think we've kind of covered most of these questions. What else do you think?
Rabbi Finkelstein,
Yes, Patty.
this is Patty. If you've taken the Moderna, is it necessary to take the Moderna in the second dose also? If you've taken the Pfizer, do you take the Pfizer in the second dose? Does it matter?
So the way that they were studied is that you take the same vaccine twice. And that's what we want you to do. It's possible that you could do Moderna than Pfizer, but we have to study it first to make sure it works. So, I definitely take the same vaccine for both shots.
We're coming up on the hour. Any other last questions? We'll take maybe one or two. Yes, go ahead. Elaine.
Somebody became anaphylaxis to a medication such as Cipro, clindamycin, Cleocin, fluoroquinolones. Is it safe to take the vaccine?
So what we suggest is people that that develop anaphylaxis easily, who use EpiPens, they need to be more careful. We still give them the vaccine. We just watch them a little bit longer. We keep epinephrine and medicine nearby just in case. As I said before, it's 1 in 100,000 people, and cephalosporin and pen allergies are 1% of the population or so. So it doesn't mean, oh, I had an allergy to a cephalosporin, I can't take the vaccine. It just means let your doctor know, and they'll watch you a little closer for a little longer.
Thank you.
So is that a big deal if someone has I can't pronounce that, the reaction, the allergic reaction, and then they get their epinephrine shot? Is that a big trauma for them to have to go through that?
Nothing is fun when it comes to medicine. You want to avoid it if you can, but it's so rare. You just can't say anybody with any allergy shouldn't take the vaccine.
It's worth it. It's worth it to give it a shot.
Yeah.
Rabbi,
Yes.
is the first dose and the second dose the same?
Yeah, they're exactly the same.
Okay.
Rabbi, can I continue my question? I read
Sure.
that if anyone took, I don't have it absolutely correct, polyethylene glycol, if they became anaphylaxis, they should not take any compound that has that.
Oh God, get it.
So the problem is that everything you put on your body, or almost everything, has polyethylene glycol in it. Cosmetics are full of PEG. Many other foods, medicines, topical things are full of PEG. Some people think the PEG might be involved in the anaphylactoid reaction. I don't think it is, but we have to study that.
Thank you.
You can't avoid PEG.
All right. So, Dr. Drew, anything that we didn't cover, or that you think you didn't cover in your video presentation because it was a little earlier? Anything else that you'd like to message to us? 100 Jewish folks here, and mostly Jewish folks here on Zoom. Anything else you want to message to us before we part ways?
Sure. So the important things are, there's a couple. The first is you need to take the vaccine. We know that for young people, the risk of getting bad infection is low, 0.5% or so. But the older we get, the more medical complications we get, the mortality can be upwards of 25%. So there's a big risk for many people. The vaccine is safe. What's great, and this surprised everybody, the vaccine is very effective in elderly people. Everybody was worried about the flu vaccine that isn't great to begin with and is really poor in elderly people. And other vaccines we see just don't work as well in elderly. This vaccine works very well in elderly people. And those are the people that are most important to get vaccinated. So the vaccine is safe, you need to take it. The other thing is we still need to wear masks and socially distance until we can vaccinate enough of the population that the pandemic will wind down on its own. So just because you had the vaccine doesn't mean you can stop wearing a mask.
And if you don't get the vaccine, you're not protected.
Right.
Yes. Finally, any thoughts on what we can do with a certain swath of the population? I meet many of them at the hospital, and nurses, all kinds of people who say they're not going to be guinea pigs for this, and it's too fast, and they're going to wait, and it seems to be slowing down the whole rollout. Any thoughts on how we can effectively deal with this problem?
Yeah, I mean, I don't think anybody knows. I mean, I spend a lot of time talking to nursing home facility workers who don't want to get the vaccine. There are certain populations that are more resistant than others, and I try to spend time talking to them, explaining to them that the vaccine is safe, it's effective, and they need to take it. That's a public health problem, and we need the public health system of the government and the universities and the hospitals to spend their time explaining to people to take the vaccine.
Sometimes it's just very hard for people to believe facts if it's inconsistent with a mindset. That's complicated. If they don't take the vaccine, they will remain at risk.
Thank you. Well, thank you both for joining us. We're so honored that you joined us. It's been so great. What an incredible thing to even know you and to be connected with you and to spend some time talking to you today. God bless you for everything you've done, and may you continue your research and may continue to knock off and tick off other diseases as well as you continue with your important research. So thank you so much on behalf of the Baron Hirsch, the Beth Shalom, the Dor Chadash, Jewish Family Service. Thank you for joining us today. And thank you all, your cousins and everybody else for joining us today as well.
Thank you.
Thank you
Have a good day.
so much.
Thank you. We applaud you.
Yay.
Move it on so I can see.
Thanks for sharing.
Thank you.
Thank you. Thank you. Thank you.
Nice.
Any last
Thank
comments from cousins or anybody else?
Let's go see who's here. Oh, yeah, we're done.
Okay. All the best, everyone. Take care, everyone. Have a great day. Thank you so much. Mary Ellen, great to see you. Any last comments from Mary Ellen?
Oh, I'm done, but thank you.
Thank you. Thank you. Great to see you.
Bye-bye.
Okay, take care. Be well.